Okay, and thank you all for taking part in this excellent conference and for coming to hear my talk. Okay, so talk to you today about a program that we have at Invenra, which we're really excited by. It's an immuno-oncology program that has some fantastic preclinical data, and we think we may have solved one of the problems in immuno-oncology using a bispecific approach to therapy. So first I'll tell you a little bit about Invenra and our technology, and then describe the molecule that we've designed and provide you some of the in vivo data that we have to support the approach that we're using. So Invenra is a small but growing biotech firm in Madison, Wisconsin, which is a beautiful northern Midwestern city situated between two lovely lakes. And it's a really great place to be a biotech because the University of Wisconsin is one of the nation's great research institutes. And so we have a lot of really good collaborations going on there, and have a really alive and invigorating intellectual atmosphere in the city. On the other hand, it's a whole lot cheaper than Cambridge. So we have complete capabilities for generating monoclonal antibodies, VHHs, and we have a bispecific antibody platform, which I'll tell you about briefly in a moment. So what we're trying to do with our bispecific platform is generate antibodies that can modulate the tumor microenvironment and exploit that to generate first-in-class therapeutics. So our platform, very briefly, looks like this. And you look at this and you say, well, that looks like an antibody, and that's exactly right. The platform is designed to give us antibody-like molecules that contain all the desired properties of effector function, half-life, stability, ease of production, purification, manufacturability, stability, all of those things, yet give us two different specificities on the two different arms. And one of the things that really differentiates us is that our platform allows us to create these molecules in small scale, high throughput, single step purification approach. This allows us to screen our molecules in their final format. And that's really critical with bispecifics. Unlike antibodies, where what really matters is, does it bind tightly and does it inhibit, or something like that? That's really what's usually important. With a bispecific, you're using the geometry to create some sort of biological effect. And therefore, the geometry can be critical. The epitope can be critical, not just the epitope in terms of the patch it binds to, but the epitope in terms of the actual way, the orientation of the variable domain versus the target can be critical for whether it works or not. So if we were to be screening each of these two binders separately and then put them together at the end, expecting it to work, we would probably be disappointed. What we can instead do is select a whole bunch of binders to one target, a whole bunch to another target, and create a matrix where we can screen hundreds or even thousands of combinations for the one that has the best biological activity. And part of this is because the light chain pairing is maintained as correct. We don't have mispairing because we've swapped out the wild type CLCH1 with a different pair of heavy domain, constant domain molecules that do not interact with CL and CH1. So you can't really have the light chain go to the wrong antibody. So this gives us a way of generating bispecific antibodies that may have complex biological effects. You can then expand these by adding an extra Fab in different locations to make bispecifics or even trispecifics. We aren't going to get into that in this talk, but it's part of the platform, is the ability to change the valency, so you have a bivalent binder and a monovalent binder. Our pipeline has some internal assets which are either immuno-oncology or anti-tumor, using different approaches. And the sniper approach is what I'll be telling you about today, uses bispecifics to increase the specificity of the antibody for a particular type of cell. And the agonist approach uses a clustering approach to improve the agonistic activity of the molecule. I won't tell you about this, but this is also a very powerful way of using bispecifics. We also have a very fruitful collaboration with Exelixis, where we are working on a number of undisclosed targets with them. And we have other partnerships and collaborations ongoing as well. So we're a very a vital company. We have a lot going on, and we're always interested in talking to people about science and about how to make better therapeutics. So, okay, so getting into antibody specificity and why you use a sniper. So antibodies are fantastically specific on their own. They can bind a target, and in most cases don't bind anything else. They can also bind with extremely high affinity, as we know. So this has made them very powerful drugs, and gives them a significant advantage over the small molecules. But it's really difficult to find a target that is expressed only where you want your antibody to go, let's say a tumor, and not anywhere else in the cell. I mean, tumor cells are, of course, human cells that have gone awry, but they produce essentially human proteins, although there may be mutations in them. But it's very difficult to find a protein that is produced only on the tumor cell and not on any other cell. So what the sniper does is it allows us to improve the specificity of binding to the cells by taking advantage of having two different targets. And so here's an illustration of how this works. So in the if you have a target cell that has these red targets and these blue targets, and a non-target cell expresses maybe just the red one, a monoclonal antibody will bind the target cells, binding to the red ones there, and also to the non-target cell, binding the red ones here. So you can get off tumor on target interactions and effects. And this is what is called a side effect. It's can often be quite toxic and can lead to a limiting of dose, limiting of efficacy, or just complete failure of a molecule. It is, of course, really difficult to find out if you're going to have this kind of issue, just because people are different. Not every person has exactly the same expression of every protein everywhere. So this is a big challenge in monoclonal antibodies. The Invenra sniper will bind only the target cell if we can find two targets that are expressed on that cell and that are not expressed together on other cells. And this is a much easier problem to solve, even though it's more complex. It's more likely that you can find a pair that is expressed only on the type of cell you want to go for. So here in the illustration, your antibody has one arm to the blue and one arm to the red, and it's binding very nicely to the target cell. Yet a non-target cell expressing only the red, or if it were expressing only the blue, there would only be one arm. And you say, well, one arm should still be able to bind, and that's true. So we get around this by modulating the affinities of the two arms so that monovalent binding is a weak binding. It comes off quickly. You don't collect very much on a non-target cell, and so it does not have any effect. And that's where I'm saying that the epitope is critical, because to create a molecule like this that has a lot higher affinity to the target cells versus the non-target, the two epitopes have to work very well together so that the antibody there's no much energy barrier to the antibody binding to both targets on the cell. Okay, so the molecule I want to tell you about today, Invenra 321, is a tumor-specific Treg depleter. Now, Tregs are thought to have a very strong effect, a real profound effect on the immune response. In general, they're one of the important ways that the immune system regulates itself, and tumors can take advantage of that by boosting Tregs in the tumor, therefore regulating the immune response out of existence in the tumor, allowing the tumor to grow. So when you have patients' tumors, you characterize them. If they are rich in T cells, they still may have an immunosuppressive microenvironment, and very often that is caused by Tregs or suppressor cells. So this is the target population that we would like to target. It's actually quite a large population, and it's a frustrating one because there's lots of T cells, but the standard immuno-oncology therapies just fail. And we think that very often it is because of the Tregs. So we are hypothesizing that if we can eliminate those tumor Tregs, this can reduce that suppressive tumor microenvironment, rebalance the immune response, and what we would like to do is target only the Tregs that are in the tumor. And why is that? It's because if you target Tregs everywhere, the immune system will end up overactive, and you may be able to cure the cancer at the cost of very serious autoimmune disease, which is not a great trade-off, and we'd really like to avoid that. So we believe that this is a therapy that, while the data I will show you is as a monotherapy and it's quite effective, that it will be particularly effective as a therapy to go along with other immuno-oncological therapeutics. Okay, so challenge with Treg depletion using monoclonals. If we look at common targets in the potential Treg depleting targets, only the CTLA-4 has shown itself to be clinically effective. It does not have an effect on peripheral immune cells, no depletion that we can see, but it has a lot of autoimmunity. Of these others, they either did not show clinical efficacy or has not been demonstrated yet, but there's some toxicity in some of them as well, and the ones there's no tox, there's no efficacy. So the field is still looking for an effective molecule. So the risks with these monoclonals, which is how most people have been attacking this, is that you'll end up depleting other cells because all of these targets are expressed on multiple types of immune cells. So we think the sniper approach can give us not only Treg depletion, but specificity for just the right Tregs, a target-specific mechanism of action, higher efficacy, and lower tox. Next problem we had to overcome is that a lot of these targets are not the same between mouse and human. And given what I was saying about the importance of the epitope, it is very hard to find cross-reactive molecules. So the approach we took was to start with a surrogate and make a mouse molecule that was effective in the mouse, and then we could try to make a human molecule that is as close to the mouse molecule as possible in terms of epitope, affinity, cell binding, effects on cells, and in every way that we can measure before going into clinical trials. So we wanted weak monovalent affinity to two of the targets that are expressed on Tregs in the tumor microenvironment. We actually used human samples to test what was being expressed on Tregs in the periphery and in tumors in human cancer patients, and found a pair of targets that was expressed overwhelmingly in tumors and in very few peripheral Tregs. So we wanted to have a strong presence of both targets and make sure that we block them only when both are present, so that the potency, monovalent potency, was weak enough that they would only bind when both are present. So we would then use this surrogate to design the human lead program. Okay, here's our first data. We made it such a molecule, and we gave it to mice with the CT26 syngeneic model. And we gave 3 different doses, 0.4 mg per kg, which is quite low for these sorts of molecules, 2 mg per kg and 10 mg per kg, which is a more normal amount. And there were 5 mice in each group, and all 5 mice were cured in 10 mg per kg, all 5 mice in 2 mg per kg, 4 out of 5 mice were cured in 0.4 mg per kg. This was just a single dose, and even a single dose of 0.4 mg per kg was quite effective. We then wanted to just find out if these responses were stable, long-lasting immune system changes to the immune system. So we took these same mice and let them grow for a number of weeks, and then challenged them. And we challenged them with 2 different tumors. We challenged them with the same CT26 on one flank and with a new tumor in the other flank. The new tumor is shown in gray, and after the re-challenge, and that grew out just fine. The CT26 re-challenge here in the 0.4 mg per kg, the 4 out of the 5 cured mice that had survived were challenged, and they all remained tumor-free. The 10 mg per kg, all 5 remained tumor-free, and of the 2 mg per kg, 1 of the 5 was able to, the tumor was able to escape and grow back. So out of 14 mice that had survived the original tumor, 13 of them rejected the re-challenge. So this demonstrates that this is producing a immune response that is long-lasting and meaningful, not just a temporary change in the immune system. So what happens to tumor growth during this period? So here in the top left, we're looking at the percentage of Tregs among the total tumor infiltrating lymphocytes. And this dotted line shows where the Tregs are around 8% in the vehicle, and it's fairly stable. In the mice that we dosed, you can see the Tregs are right down at the bottom, even up to 12 days later, which is the antibody, there's not much antibody left almost, yet the T effectors, the CD4 and CD8 T effectors start out, the Tregs are not what is going to actually kill the tumor. It's the lack of Tregs that allow this boost in effector that's going to kill the tumor. So that's what's happening around day 12 or so.
You're no longer sharing your screen.
Okay, is that now sharing?
Yes.
Yes.
Okay, and should I go over this slide once more? This is a very important slide. Did it drop out through it?
That's when it dropped out, yes.
Okay, I'll go through this slide once more. So this is what happens during the first 12 days after treatment. We measured the tumor Tregs and the tumor T effector cells at days 5, 9, and 12. In the vehicle-treated animals, the Tregs were around 8% of all tumor-infiltrating lymphocytes. When we measured Tregs in the tumors of the treated animals, they were almost unmeasurably. They were down below 1%. The effector cells, on the other hand, were not much changed until day 12, when they all of a sudden spiked up to a very high percentage of the TILs. So, 15 to 20% of TILs were CD8s and 15 to 20% were CD4 T effectors. If you look at the growth of the tumor during that period, you see that the tumors of the treated groups grew quite well up till about day 12, and then they started shrinking. So this suggests that the treatment is reducing Tregs right away, but that it takes some amount of time for the T effectors to then come up and start actually killing the tumor. So it's a delayed response. But remember, this is a single treatment at day 0, and even day 12, there's no Tregs in the tumor. So it seems to have a profound and long-lasting effect. Meanwhile, if you look at the periphery, the Tregs are in the bottom here, around 1% in the treated and 1% in the vehicle. There's essentially no difference between the two. And the CD8s and CD4s also are both within kind of the normal range with the treated and vehicle. So what this really shows is that our drug is going to the tumor and working there, but not having very much effect on the periphery. So we believe that this model can remodel the tumor microenvironment, be favorable for T cells and T cell therapies. So we were feeling like we really wanted to challenge this more, given that it was so effective. So we tried it with tumors that were larger, and if we started with larger tumors, the vehicle, of course, they grew out, and the sniper, 4 out of 5 here were cured with a single 2 mg per kg dose, starting with tumors that are greater than 100 millimeters cubed. So our next goal was to really compare how well this antibody works compared with other Treg depleting antibodies. And the only ones we have data on so far is CTLA-4, this sort of standard Yervoy type antibody. This is the mouse version. And we wanted to compete head to head with the CTLA-4 antibody. And we'd like to eventually go against all of these different antibodies. So here's some data comparing Invenra 321 to an anti-CTLA-4 mirroring surrogate in a CT26 model. In this particular case, the Invenra molecule with 1 dose of 2 mg per kg cured 3 out of 5, with a dose of 0.4 cured 1 out of 5, and a dose of 0.08 cured 2 out of 5. So the curing doesn't seem to be very dose dependent. It's probably dependent on the immune system of the mouse, but even down to 0.08, it's showing significant efficacy. The CTLA-4, on the other hand, showed 1 out of 5 at the highest dose of 2 mg per kg and no cures at all at the lower doses. So that suggests that we're more potent than CTLA-4 molecules. We then went to a more challenging tumor model, the Renka renal cancer model, and found that at 2 mg per kg, 4 out of 6 complete responses, with CTLA-4 was getting no responses at 2 mg per kg. But to be really fair, we thought, well, let's compare our molecule at single dose of 2 mg per kg with the more standard CTLA-4 treatment regimen, which is 10 mg per kg 3 times every 2 days. So with that 15 times more dose total, 30 mg per kg versus 2 mg per kg, 2 of the mice were cured. So it has some efficacy, but a lot lower efficacy, even with 15 times more drug being delivered. So we've shown a lot more potency. And with a larger tumor model, a larger Renka model, we were unable to cure the mice, but there was a lot more delay in their tumor growth compared with the 99, which is the CTLA-4, even given at 10 mg per kg. And we were giving the sniper at 2 mg per kg. So you can just see the survival curve is significantly stretched out with the sniper and only a small amount stretched out with the CTLA-4 binder. Okay, so what can we say about what's going on in the tumor? We did some immunophenotyping, and here is at day 7 in the tumor. We found that there was not much difference between the vehicle and the sniper and 99, the CTLA-4 binder, in CT4 positive cells total or in effector cells. And but in Tregs, the sniper was down more than the other two. Effector cells, the sniper and 99 really had no effect, no differential effect. They both were somewhat better than vehicle. Remember that from days 5 and 9, there wasn't very much going on that we had seen in the tumor. So then if we go to day 12, the situation changes. Okay, with the CD4 total number of cells is similar, but the effector cells with the sniper is way high, and with 99, effector cells are now very low. And in contrast, Tregs are very low with the sniper and very high with the 99. So we believe that this is actually giving us a much more effective depletion of T cells. Similarly, with CD8 cells, the CD8 effector cells are greatly enhanced with our molecule, and you can't even find them with the 99. So we think we have a molecule it's really exciting. We think it has the possibility of having minimal toxicity and maximal efficacy, be able to be in combination with things like PD-1 inhibitors or other immuno-oncology therapies, and really be able to cure a lot of people of cancer. So our next steps here is we're working on additional efficacy studies and starting humanly development of the human version of this. So with that, I'd like to thank the Invenra team, which is bigger than this, but we couldn't take a picture showing our lovely faces this year. It would have to just be all masks. So this is the group from last year. And our collaborator, Paul Sondel, in his lab at University of Wisconsin, who have been really great with this. And with that, I'm happy to take questions. We have a few minutes left.
Thank you very much, Dr. Davis, for your amazing presentation and showcasing your company's platform, which is quite interesting. If there's any question for Dr. Davis, you can type it and we can answer accordingly. Oh, there's just one question that came in from Hugo, CEO of PhenoSwitch. And he's asking, antibody engineering, including bispecific antibody, are indeed wonderful tools to increase treatment specificity. However, the production quality and reproducibility of the bispecific can be quite challenging. How do you tackle this in your platform?
Well, that question is a whole talk in itself. It's a great question, but the bottom line is that our platform allows the molecules to assemble themselves very well straight out of the cell. So we get a high yield of correctly assembled molecules, and purification is not generally any more difficult than it is with a regular antibody to polish it up. Our yields are excellent. Just with a transient transfection, we often get 200 or 300 mg per liter yield of molecules without any optimization of anything. So it seems to be a very robust platform and amenable to manufacturing.
Is there any other?
I have a brief question. So when you take this into humans, you were ending up showing some differences in response depending on the tumor that you used. Do you envision having to create different snipers for each tumor type to be able to ensure the specificity, or have you identified something that can be generalizable to a broad range of tumors?
That's an interesting question. We believe this is generalizable because it is aimed at an immune system molecule, not a tumor molecule. So different tumors won't directly drive those responses. You know, they can't just A tumor itself can change the expression of one or another surface marker, but it's much more indirect with immune system cells. So from what we can tell, because when we did the original work of searching for a pair of targets, we found that if we used different types of tumors, it wasn't just one sort of tumors from patients. And the different types of tumors all showed this pattern of these two targets being present in the tumor and not in the periphery. So we think it's going to be broadly applicable. I can't say universal, nothing's universal in cancer, but we shouldn't need to make a different molecule for different cancers. I think any cancer that has the Treg problem, we should be able to address.
All righty. Well, thank you very much for a great talk.
Yeah.